Water filters light. In the ocean, an organism's depth dictates the behavior of photons, making light manipulation the primary driver of survival. To navigate this medium, aquatic life has optimized two drastically opposed evolutionary strategies: maximizing the reflection of incoming light or absorbing it completely. In the shallow, sunlit Amazon basin, the freshwater angelfish exemplifies the first strategy. It uses highly reflective scales and high-contrast pigmentation to signal conspecifics. Thousands of feet down in the North Pacific, the Pacific Viperfish, Teleost macuni, executes the exact opposite approach. Operating in the crushing pressure of the bathypelagic zone, it relies on total light absorption to maintain predatory stealth. Despite their extreme environmental differences, both of these Teleost fishes share a severe biological limitation. Neither species possesses the internal enzymatic pathways required to synthesize primary color pigments from scratch. Without the ability to manufacture their own biological colorants, their physical appearance is strictly bound to the chemical compounds they extract from their diet. The biological conflict is immediate. These organisms must manipulate environmental photons to survive, yet they lack the internal capacity to generate the required pigments. Because they cannot synthesize these molecules, a fish's optical properties and its ultimate viability as a species are dictated by a combination of dietary bioaccumulation and specialized anatomical engineering. To achieve vivid coloration, the angelfish utilizes a highly specialized dermal architecture. The outermost epidermal layer of its skin is entirely transparent. This clear window exposes specialized pigment-bearing cells, called chromatophores, resting in the dermis below. The actual pigment inside these cells is beta-carotene, an exogenous molecule composed of 40 carbon atoms arranged in a long horizontal hydrocarbon chain. The critical feature of this molecule is its series of conjugated double bonds stretching across the carbon skeleton. These bonds act as a highly tuned antenna for interacting with incoming photons. Due to its specific chemical geometry, beta-carotene heavily absorbs wavelengths between 400 and 500 nanometers. This corresponds exactly to the blue and green portions of the visible light spectrum. By trapping the blue and green light, the molecule forces the remaining wavelengths to scatter outward. This actively reflects the red, orange, and yellow spectrums back to the observer. Only plants, algae, and certain microorganisms can construct this hydrocarbon chain through a process called the isoprenoid pathway. Higher animals simply lack the enzymes for this synthesis. Consequently, the angelfish must consume its color. Once digested, the lipid-soluble beta-carotene molecules are absorbed, transported through the bloodstream, and deposited directly into a specific class of chromatophores known as xanthophores. The vivid orange and yellow hues of a shallow water teleost are a direct physical manifestation of its dietary bioaccumulation. To measure exactly how heavily this dietary intake dictates physical appearance, researchers in 2009 isolated beta-carotene dosage as a strict variable, tracking the physiological changes in angelfish over a 75-day period. This bar chart displays the baseline data for the experiment. Fish fed a control diet without beta-carotene supplements recorded a total internal carotenoid concentration of just 4.29 micrograms per gram of tissue. But tracking the peak response reveals a massive multiplier effect. When researchers spiked the feed with 150 milligrams per kilogram of synthetic beta-carotene, the internal concentration in the fish surged to 15.77 micrograms per gram, nearly quadrupling the baseline. This accumulated pigment did not distribute evenly throughout the body. The carotenoids actively concentrated at significantly higher densities in the skin than in the muscle tissue, maximizing the external visual output. Beyond surface coloration, these lipid-soluble molecules also embed themselves into cellular membranes, where they act as powerful antioxidants to quench free radicals and enhance overall immune response. However, the data revealed a significant physical trade-off. The 150 milligram synthetic dose successfully maximized visual reflection, but it failed to produce the healthiest overall organism. A separate control group fed exclusively on live tubifex worms naturally developed a slightly lower pigment concentration, yet they recorded the maximum specific growth rate and the highest overall survival percentage. While exogenous molecular additives can artificially maximize pigment bioaccumulation and physical reflection,
A single chemical isolate cannot replace the complex nutritional profile required to drive true biological health.
Descending back down into the deep ocean, the Pacific Viperfish faces an entirely different set of physical laws. In the bathypelagic zone, sunlight is entirely absent, and hydrostatic pressure is crushing. Here, reflecting light is a lethal error. The primary optical objective is achieving a zero reflectance phenotype to avoid illumination by the ambient bioluminescence of other deep-sea predators. Instead of transparent scales displaying pigment, the viperfish relies on ultra-black skin. The dermis is saturated with microscopic, heavily pigmented organelles called melanosomes, tightly packed into a continuous layer. When stray bioluminescent photons hit this surface, the optimized geometry of the melanosomes forces the light to scatter laterally. The photons bounce endlessly between the structures, transferring their energy until they are fully absorbed, reducing reflection to near absolute zero. With external light neutralized, the viperfish deploys its own active photon generation. It utilizes specialized light-producing organs called photophores, prominently located on an elongated dorsal fin ray and in rows along its ventral side. This blue bioluminescence serves two precise ecological functions. The flickering dorsal light acts as a targeted lure to draw in prey, while the ventral rows provide counter-illumination camouflage, masking the fish's silhouette from anything looking up from below. The intensity of this light is biochemically controlled by adrenaline levels within the fish. To detect the faint movements of incoming prey drawn to the lure, the viperfish relies on heavily modified optical rods packed with rhodopsin, a transmembrane protein hyper-tuned to low-light capture. Deep-sea stealth requires the complete abandonment of dietary reflection. The fish must instead evolve its skin into a highly engineered physical photon trap. Weaponizing light to lure prey in darkness has severe biomechanical consequences. The viperfish transitions instantly from motionless camouflage into violent kinetic acceleration to strike. This high-speed intercept, occurring in a medium where light provides no directional contrast, necessitates a specialized cranial structure, the loosejaw adaptation. The skull is hinged, allowing the entire upper cranium to rotate backwards, while the lower jaw drops, facilitating the capture of prey that would otherwise be impossibly large for the predator's body size. Its fangs are abnormally long, so massive they rest outside the mouth, near the eyes. This specific placement ensures the teeth act as forward-facing spears, impaling the target upon the initial high-speed impact. To survive the kinetic force of these collisions, the viperfish possesses a built-in shock absorber. The first two vertebral centra behind the skull are replaced by an enlarged epineural ossification, cushioning the central nervous system against the violent impact. This aggressive motility creates a critical hydrostatic problem. A traditional gas-filled swim bladder, used by most fish to maintain depth, would fatally expand or rupture during rapid vertical strikes in the high-pressure bathypelagic zone. To resolve this, the viperfish lacks a swim bladder entirely. It achieves neutral buoyancy through a biochemical alternative, suspending massive amounts of acidic glycosaminoglycans within a gelatinous tissue matrix. The ecological choice to weaponize light in absolute darkness forced a cascade of anatomical compromises, necessitating a complete evolutionary redesign of the animal's skeletal and hydrostatic biomechanics. Despite occupying opposite ends of the pressure and light spectrums, the angelfish and the viperfish are bound by the exact same biological constraint, an absolute inability to synthesize primary pigments. The angelfish resolves this limitation by acting as a biological accumulator. It extracts beta-carotene from its environment, converting exogenous dietary chemistry into active optical reflection and internal cellular defense. The viperfish abandons dietary color altogether. It relies exclusively on intrinsic anatomical engineering, deploying dense melanosome structures to physically trap photons and ensure absolute stealth. One organism engineers its surface through the chemical accumulation of molecules. The other engineers its surface through the physical geometry of its tissues. Both are distinct solutions to the exact same evolutionary imperative. The need to control the physical behavior of light forces life to carve out radically divergent pathways for survival. Whether weaponizing the absence of light in the depths or reflecting its abundance in the shallows, these fish have optimized their entire biology to master the specific optical physics of their environment.